Back on Neuro Insights, I'm Rittika. This episode focuses on mitochondrial dysfunction in amyotrophic lateral sclerosis, also known as ALS, where energy deficit and oxidative damage drive motor neuron demise. With us is Kanak Chaudhary, Master of Science in Biotech, drawing from her cellular metabolism knowledge. Kanak, welcome. I would like to ask you a question. So could you elucidate how mutations in SOD1 and C9orf72 disrupt mitochondrial bioenergetics and dynamics in ALS pathogenesis?
Sure, thank you so much for having me again. The mutations in SOD1 lead to misfolded proteins that accumulate on the outer mitochondrial membrane, impairing the respiration and calcium buffering. And the other one, that is C9orf72 expansions, add another layer into it, disrupting the mitochondrial trafficking along axons and altering interactions with the endoplasmic reticulum. Together, these mutations compromise ATP production, elevate oxidative stress, and destabilizes the fission-fusion balance, all of which make the motor neurons highly energy-dependent cells, especially in the case of ALS.
Right. So there was a very good explanation on the core mechanisms here. What I think is very challenging is the fission fusion imbalances. Now, mitochondrial fission via DRP1 and fusion via MFN2 are dysregulated in ALS. So, what live cell imaging with super resolution microscopy shows about these processes in motor neurons? And how do they link to axonal transport failures?
Super resolution live cell imaging has given us the striking views of mitochondrial dynamics in the case of ALS motor neurons. Uh, we see excessive fragmentation from hyperactivated DRP1 and reduced fusion due to the loss of MFN2 function. This imbalance interrupts the long mitochondrial networks needed to fuel axonal transport. As a result, mitochondria stall, fail to reach the synaptic terminals, and starve the neurons of energy where it's needed the most, directly linking dynamics defects to transport failure and synaptic uh, degeneration.
That was indeed a very, very detailed answer. Now, going to oxidative stress amplification, which I think is a main root issue. So, like reactive oxygen species or ROS from dysfunctional mitochondria forms vicious cycles. And how do NRF2 RE pathways activation attempts the the compensation rather, and what cutting edge antioxidants like edaravone variants target specific ROS sources in clinical ALS subsets.
The dysfunctional mitochondria overproduce these ROS, which in turn damage the mitochondrial DNA and proteins, creating a vicious cycle. The NRF2 air pathway acts as a compensatory shield by inducing antioxidant enzymes, but in ALS, its activation is often too late. New antioxidant strategies go beyond uh, general scavenging, which has different variants, and mitochondrial targeted antioxidants like MitoQ and SS31 are designed to neutralize these ROS at their source, showing promise in certain patient subsets by protecting mitochondria directly.
So these are extremely critical cycles. Now let's move on to glial contributions. So astrocytes and microglia exacerbate mitochondrial defects via inflammatory signaling. What single-nucleus transcriptomics reveal about non-cell autonomous effects? Okay, I'll just repeat that again. Astrocytes and microglia exacerbate mitochondrial defects via inflammatory signaling. What single-nucleus transcriptomics reveal about non-cell autonomous effects? And how do mitochondrial transfer via tunneling nanotubes offer therapeutic responses and potentials?
So the astrocytes and microglia add a non-cell autonomous layer to these mitochondrial stress. And single nucleus transcriptomics reveals that these glia upregulated the inflammatory genes and secrete toxic mediators that further impair the neuronal mitochondria. Interestingly, the therapeutic counterpart is mitochondrial transfer, in which the astrocytes can deliver the functional mitochondria to stressed neuronal and neurons that are there through the tunneling nanotubes. This natural rescue mechanism now being studied experimentally and could be harnessed to restore the bioenergetics in ALS.
Right. So let's move into genetic modifiers. So TDP-43 pathology intersects with mitochondrial RNA processing. How do these alter mtDNA stability, and what CRISPR-based screening identify enhancers or suppressors for mitigating dysfunction in ALS models?
So the TDP-43 pathology interferes in the mitochondrial RNA processing, leading to incomplete transcripts for respiratory chain proteins and destabilization of mitochondrial DNA. This undermines the overactive and oxidative phosphorylation capacity in the CRISPR-based genetic screens in ALS models are beginning to identify modifiers that are the genes that either worsen or suppress these effects and the defects. For example, certain RNA binding proteins and mitochondrial quality control genes emerge as potential intervention points to buffer against these TDP-43 driven mitochondrial collapse.
Right, those are some really advanced tools. Let's move on to metabolic reprogramming. Shifting from glycolysis to oxidative phosphorylation is impaired in ALS. What metabolomics profiling in patient biofluidics suggests about ketone bodies in alternative fuels, and how could gene-edited stem cell therapies restore metabolic flexibility?
So the ALS neurons often fail to shift the flexibility between glycolysis and oxidative phosphorylation, leaving them metabolically rigid. And metabolomics profiling of patient CSF and plasma highlights reduced glycolytic intermediates and compensatory increases in ketone body utilization. This has inspired the trials for the and ketogenic strategies to supply the alternative fuel. At the same time, gene edited stem cell derived motor neurons offer a way to restore metabolic flexibility by correcting the intrinsic mitochondrial defects before the transplantation and opening a regenerative route to support the energy balance that is disturbed.
These are indeed very promising directions. So AI model predicting mitochondrial trajectories from multiomics data are an emergent concept. So, how might they guide precision medicine in sporadic ALS, and what challenges in biomarker validation persist for early detection?
As the AI-driven models are now integrating the genomics, metabolomics, and imaging data to predict how mitochondrial dysfunction evolves in sporadic ALS, these models could help the stratify patients uh, early and tailor interventions to their specific mitochondrial trajectory. The challenge lies in biomarker validation. While we can measure the signatures like altered acyl carnitine or the mitochondrial DNA fragments, we still need consistent and clinically reliable markers for early detection. And bridging that gap will be critical to making AI guided precise medicine that is a reality in ALS.
Kanak, thank you for the deep dive into ALS mitochondrial rules. Thanks. Please, listeners, stay tuned for the next episode.